Good afternoon. My name is Johan Van Hoof. I'm heading the Janssen vaccines R&D organization, and it's a real pleasure to be here today at Future Human to present you an update on our COVID vaccine program. As shown on the second slide, it's important to remember everyone that we are talking about an R&D project, so in no way I want to suggest that this vaccine candidate has already proven efficacy in human, and we will need to weigh the outcome of the data late this year, early next year, before we know with certainty whether this vaccine is efficacious, yes or no. Janssen is already in Ireland since more than 85 years. We employ more than 5,000 skilled employees across Ireland with 4 major manufacturing sites, and we are active in the 3 sectors: in the medical device sector, in the consumer sector, and the pharmaceutical sector. Today, I will provide you an overview of where we are with our COVID vaccine. Needless to say that this virus is raging throughout the world. Today, we have more than 41 million cases reported worldwide, and it's certainly a large underestimate, and more than 1 million people have died. Looking at the virus at slide 5, the COVID, coronavirus disease, is caused by what is called now the SARS-2 virus, which is named after the first SARS virus that was identified back in the beginning of this century. There's a lot of similarity between that virus and the current virus, and that's also why we are able to leverage the lessons learned from that experience. The virus uses its spike protein to attach itself to the ACE2 receptor in the human cells, specifically in the lower respiratory tract. And we know that if you induce immune responses, neutralizing titers, neutralizing antibodies against the spike protein, at least for the first SARS virus in animal models, you could protect against infection. You can neutralize the virus. The hypothesis is that for the current virus, if you induce such neutralizing antibodies and cell-mediated immunity, you will have the same result. At the same time, we also know from the lessons from SARS-1 that you have to induce what are called Th1 responses. You should avoid Th2 responses, and this to avoid the phenomenon of enhanced disease. Within Janssen, we are using 2 manufacturing platforms, of which the key features are very exciting in context of a COVID vaccine. We are using the adenovector, which is a non-replicating adenovirus, which is genetically engineered, in which we can put transgenes depending on the disease we want to prevent, whether it's HIV, RSV, Ebola, Zika, or COVID. Today, we have vaccinated in our R&D programs more than 100,000 people. On this slide, it still mentioned 67,000, it's more than 100,000. That have been studied in clinical trials, and throughout those studies we have seen a very acceptable tolerability profile, and we have not observed any significant safety signal. We also have observed very clear Th1 responses, which is very good to reduce the risk of enhanced respiratory disease, and it's a platform that has been fully upscaled now to 1,000 liter, using a manufacturing cell line that can be grown at very high cell density, and thus promising very high yields and enabling us to produce big amounts of quantities in a short term, in a relatively short term. We also see that this vector has favorable thermal stability at 2 to 8 degrees, which allows distribution using the conventional vaccine distribution channels. It is clear, as illustrated in slide 7, that collaboration is important in this type of undertakings. We are collaborating with many partners. We have announced early this year already that we are working with BARDA in co-funding this project. We are also working together with academic centers. We are also working together with contract research organizations to help us expanding our supply chain. And we are clearly also very closely interacting with many governments and health authorities worldwide. Slide 8 actually shows us the timeline since the beginning of this year. We started off in January once the genetic code was published, and we at that moment already made several candidate vaccines where we used DNA vaccines as tools, because they allow us to rank the different transgenes candidate vaccines in terms of performance in animal models. Based on that, we selected 3 candidate transgenes that we incorporated in our adenovectors and then did extensive preclinical testing to identify the most promising candidate that we included then in our adenovector, with which we started phase 1 manufacturing by the end of May, such that the phase 1 study actually could start already end of July. The first interim data after the first dose were actually collected at the beginning of September, and it is these encouraging data that allowed us to start the first efficacy study that started by late mid-September. And we soon hope to start a second efficacy study where we are looking into those schedule. So in slide 9, actually, you can see that we actually have been working a lot in parallel, which explains the fast speed, the high speed with which we can progress. We have done extensive characterization in animal models where we look at safety, immunogenicity, and also protection, while in parallel investing in manufacturing capacity, and where we have started already producing at large scale the vaccine candidate. And of course, all of this then allowed us to start the clinical testing in July. Slide 10 illustrates the 2 publications, major publications that were published this summer, and that actually reported on the results obtained in 2 important animal models. One relates to non-human primates, where we demonstrated that a single dose of a vaccine protects against infection, both at the level of the lungs, but also at the level of the nose. And we also demonstrated in hamsters that vaccinating hamsters can prevent disease symptoms, which is also an important encouraging observation. Slide 11 shows actually the key data that were published in Nature, where we see the results that were observed in terms of virus that could be isolated from vaccinated animals with the different candidates. And we look then at isolates taken from the lungs, which is called BAL, bronchoalveolar lavage, on the left, and also virus that is isolated from the nose using nasal swabs. The final candidate that is in development is the one that is called SPP here, which is these greenish lanes. And you do see that in the lungs, none of the animals that were vaccinated had still viral replication in the lungs, while the placebo ones had a very high level viral load. And you also see that at the level of the nose, out of the 6 animals, 5 animals were actually protected, and 1 animal still had a limited amount of a virus replicating in the nose, but also time-wise for a very short period. So what this says is that it seems to offer protection against the lower respiratory tract, but also at the level of the upper respiratory tract. Remains to be determined, of course, how relevant and predictive this is for the situation in humans, but certainly it's very encouraging, especially as these data were observed after the administration of a single dose. The next slide shows a correlation between antibody level and viral replication, and you do see that the higher the antibody level, the lower the viral load, and as of a certain threshold, you don't see any viral replication anymore, which is biologically, of course, very plausible. All of this led to the start of the clinical trial program, of which slide 13 offers the overview. And you see here that we, in a phase 1 study, actually looked into the safety and immunogenicity, where we looked at 2 different doses, 2 different strengths of antigen, and we're looking into the effect of a single dose administration or giving 2 doses 2 months apart. We're looking here both at the younger cohort, 18 to 55 years of age, but also at people over 65. Then there's a phase 2 study that looks at several additional questions, such as the duration of interval, the robustness of the immune memory, and all of these data will be very important to allow us final decisions on the schedule to use. And all of this then also resulted in the start of the phase 3 study in mid-September, where we actually are looking to address the question, can this vaccine prevent moderate to severe COVID disease in people who are vaccinated? And we're looking actually into people over 18 years of age, We want to include at least 30% of this population, people over 60. We also want to include those that are most vulnerable to the disease. So we are putting a lot of priority on making sure we recruit the right population, and the sample size will be up to 60,000. Later in November, we plan to start also a two-dose schedule. The two-dose schedule is important because one dose might be very useful in context of an epidemic outbreak to have early onset of protection. The two-dose schedule might potentially give additional benefit in terms of durability, and so that's why we think it's important to address both of the questions. So slide 14 actually summarizes the encouraging data that we have seen after the first dose, and on the basis of which we actually decided also with agreement from the regulators from FDA to start that study. We did see that the safety profile was very supportive for further development, although the reactogenicity was somewhat higher for the high dose in the younger groups. And as the immune responses were the same for both of the strains, we decided to start a phase 3 study with our low dose vaccine. Of course, it is important to make sure that the vaccine will be available in large amounts once efficacy is demonstrated, and that is why early onwards in the year, the corporation took the commitment to really invest at risk in large-scale manufacturing, and our ambition is to make sure that we have at least 1 billion doses of a vaccine available by the end of 2021 to be able to address the enormous needs that will be there worldwide. Finally, it is clear that this development is going at very high speed, but at the same time, we don't want to make any compromise on the quality and the safety of the product we are developing. And that is, it's in that context that our CEO, together with CEO from other pharmaceutical companies, made the pledge to make sure that we will only file this vaccine when we are convinced that we have sufficient robust data to support the safety and the effectiveness, and we will put the safety of the communities we want to vaccinate at the center of our activities. And with that, I'm concluding my talk, and I thank you very much for your attention. Thank you.